Welcome again, guys. We are talking about the microscopy lecture series, and in this video tutorial we'll be talking about the scanning tunneling microscopy. Now, scanning tunneling microscopy is one of the major advances of the change in electron microscopy, and it's a kind of different feature of microscopy that we use, because up till now we have talked about the microscopy with the light source, right? They are called the light microscopy or bright field microscopy, I can say. Then we have also seen using electron, we can actually look at very high resolution images using the scanning electron microscopy as well as the transmission electron microscopy. So it's electron as a source. Now in this case of scanning tunneling microscopy, we are also using electron as a source, but remember here the electron that we use in electron microscopy, that electron behaves like a particle, just like the classical phenomena of physics. But the electron that we use in scanning tunneling microscopy behaves like a wave. So this is called the quantum mechanics. So the way that we use, the idea, the principle that we use in scanning tunneling microscopy is about with the quantum tunneling effect, or with the quantum physics, the law of quantum physics, right? Because the electron can behave like a particle. When an electron can behave like a particle, let's say we have a barrier in both the cases. But when the electron behaves like a particle, just like the classical physics suggests us, in that case that electron, let's say, that electron will go and hit to that wall and bounce off and release. So this is a reflection if the electron behaves like a particle. But this electron, if it behaves like a wave, this electron has the capability to pass through that wall. That is the idea. So that is called as the quantum tunneling. So that's what we talk about in this video. So this is the principle that is the core foundation of quantum tunneling microscopy or scanning tunneling microscopy. Remember, STM, scanning tunneling microscopy. Why the term tunneling here? Because of the use of quantum tunneling effect. Now the quantum tunneling effect is telling us the classical picture is electron can go and attach to the energy barrier and hit the energy barrier and can return. But in case of if they behave like a wave, that wave can pass through that energy barrier from one side to another side. That is possible. That is the quantum tunneling effect. We use this quantum tunneling effect here. So the idea here, what we do in the scanning tunneling microscope is that obviously we have a sample, right? We have a sample, whatever sample let's say we have. That sample majorly we use in this case, it's not always living sample or not even living samples. We use this quantum tunneling effect or the scanning tunneling microscopy to understand the surface of different atoms, the surface of different, let's say, different molecules that we see, right? Different metals, we can see the surface of graphite, we can see the surface of gold, surface of silver, and all this, the surface of them. So how they are arranged, how they are organized. So it's not only important from the perspective of biology, but it is more important in the perspective of chemistry and physics, okay, and many more, geography also, because geology we often use scanning tunneling microscopy. Now the idea here, we have the specimen, this is the specimen, and what we also have here is the structure called the tip. The tip is nothing but it's made up with metal with high conductance, and this tip is responsible for releasing electrons from there, right? So what we actually do, we connect these two things, both of them are electron carriers. So we actually connect the tip with the sample in some way, right? So if we connect it with a particular voltage, remember, if we add a particular voltage between this tip and the specimen, electric current start to flow between them. That is the idea. Due to this voltage between the tip and sample, current waves start to flow from the tip to the sample, or from the tip to the specimen, right? So this is the idea. Now the idea here is the amount of electricity that is generated and that is formed between the tip and the sample, it is indirectly proportional to the voltage. So if you apply higher voltage, the electrons that will be released will be low, If we use lower voltage, the current flow will be high, right? So they have indirect proportionality between this voltage and the current flow, right? So if you look at in this image, we can see it pretty clearly that what we use here, the tungsten tip here, and in this case you can see this is the tip of the atom with a positive charge, and here is the sample atoms negative charge, and we create this voltage there, attach these two things to pass some light, pass some current between them. Here you can see the voltage arranged there, right? So once you do that, the idea now is to detect the surface structure of the specimen, which is this one, right? So how could you do that? Now here is the idea. Now let's say this is it, and the sample, let's say this is how the sample looks like. For example, the surface of the sample looks like ups and downs and all the way like that. Now here is the idea. We have this tip. Now the tip is attached with a rotator which can move from one place of the sample to other place of the sample, right? So during that process, we can use two important techniques to find out the surface structure of the sample. One of the technique is termed as the constant current. Another one is termed as the constant height. So constant current and constant height. The idea in case of constant current is, let's say we bring this tip here. Say this is the tip, for example, say tungsten tip. Okay, now the idea in this case is, so the idea here is that this tip is providing that current because of this voltage gradient, because of this whatever voltage gradient is present, it is transferring some of the electrons there. So as it is transferring some of the electrons during this process, so there will be a particular current that will be generated between the tip and the sample's surface, right? Now as you can see, there are two factors. One is the amount of current, another one is the distance between the tip and the surface. The idea here, if the distance between the tip and the sample surface is close, I mean they are very close to each other, in that case the current flow will be more because the distance is less. So if the distance is less, the current flow will be more, right? And the distance is high, the current flow will be less. Again, indirectly proportional in this case. So indirect proportionality again. So the idea again in this case, what we see here is that the tip, what we want to maintain is a constant current, right? So if we want to maintain a constant current, in that case we should have a constant flow of current irrespective of the distance that we see. So we need to bring this, we need to position this tip in such a way so that current remains constant throughout the scanning period, right? But the surface area is not even, it's uneven. So the idea here, the distance between the tip and the surface area point here in this case, let's say this is point A, the distance between point A and tip here is small, and there is a particular current. Now once this tip moves a little bit and the tip is now in position somewhere here, and as you can see the distance between this tip and the position B of the surface is more. So accordingly the current flow will decrease. But here we want to maintain a constant current. So to maintain the constant current, this tip will come down a little bit to compensate the distance between the tip and the point of the surface to maintain that constant current. Similarly again, when it comes down to this point of C, the distance even becomes more. So the tip should come much closer to again maintain that constant current. So to maintain the constant current throughout the surface, like uneven surface like this sample, this tip needs to come down sometimes and go up sometimes. So in this way, whenever it is coming down, whenever it is in close proximity with the sample specimen, in that case that data is fed to the detector. Detector record that the specimen is in depth there, and wherever there is a height up there, so the tip will go up. So again in that case it will record the data that yes, definitely the surface is higher, that's why the tip goes up. So whenever tip goes up in the detector, it detects the higher surface. Whenever tip goes down, it detects the lower surface. In this way we can actually construct this image by looking at the detector, kind of the similar image that we see in case of the sample. That's how we can detect using the constant current. And we can also use the constant height method. In that case, what we do, in that case we maintain the height constantly. The same thing is going to happen. If we maintain the height constantly, this tip is going to be present as it is and throughout the scanning process. But let's say here, once this tip is present to the point A, which is pretty closer, so the current flow is high in that case, very high current flow. Now, just after that, when the tip is now becoming to the point B, the current flow will be less. So what we observe in detector is a change in the current flow, because we maintain the height constant, so there won't be any change in the height. The only thing will change in this case is the current flow. So sometimes we see high current flow, sometimes we see low current flow. So if we see, now let's say, if we see the high current flow in this case, let's say in point A, current flow is very high, then again it's going down and low, then again slightly high, then low, then slightly high. So again we can construct the surface structure pretty easily. So these are the two methods of calculating. So this is the principle. As you can see here, this is the STM tip, and again these are the electrons that are coming out from the STM. This is the voltage gradient, plus here, minus here, and as it is moving in different surface, let's say the surface area is changing like this, you can see, as you can see here, difference in the graph, as you can see in detector. So detector can detect this change in distance or change in the voltage, change in the current, right? So by looking at this, we can tell what's going on. And the idea here is this tip will migrate from one place of the sample to another place of the sample, from scanning that sample from one to another place, right? So what it uses here, it uses the property of electron as a wave for this electron to be inserted to the surface of the sample, so that the current flow is possible, right? So that's how the whole process of scanning tunneling microscopy actually work. And this is the instrumentation of scanning tunneling microscopy. As you can see here, let me take another, yeah, as you can see here, this is the probe, this is the tip, this is the tip, tungsten tip usually, this is the sample, the voltage is applied between this tip and the sample, and it is being tunneled with the amplifier, because the thing is, the energy, I mean whatever surface current it generates, that current is pretty low for the detector to detect. So what we need to do, we need to amplify that signal. So for amplifying that signal coming out from the tip, we require this tunneling current amplifier, because this tunneling current amplifier will amplify the current that is coming from this tunneling effect between the tip and the sample. And we get that, and we have a control scanning unit which controls the scanning property, how this tip will move from one place of the sample to another, and then finally we can record this data using a CPU or processing unit. Okay, and this is in action how the process works, as you can see from different angles actually, but the idea here, you can actually construct this kind of beautiful images of the surface by looking. And this is the actual scanning tunneling microscope. That's how it looks like. It not at all looks like any kind of microscope that we've seen previously. So it's a microscope because it helps us to understand the structure in very fine level, because the distance between the tip and the sample is kind of in angstrom level. So it's very good actually in resolution and all these things. As you can see here, it's even more like 50 nanometer range. It's giving us that much of the detail of the structure in different layers. It's very good. But we can use it for live imaging also. We can use it for, let's say here you can see the cells, and we can also see some cilia coming out from cell surface. So we can actually use it in different purposes. But the use of scanning tunneling microscopy is not limited to biological applications. It is vastly used in many different fields of science. So that's it guys, and I hope that's helpful. If you like this video, please subscribe, hit the like button, and put some comments out there. Share this video with your friends in the social media. Thank you.